proteins and amino acids by looking at the way we think about and talk about the folding of a protein. Remember, protein's not the same as the other macromolecules. It can't just sit there as a linear molecule. It really needs to fold in order to have function. So the linear sequence of amino acids, starting at the N terminus and finishing at the C terminus, we simply call the primary structure or the primary sequence of the polypeptide. So in this case, we've got a really short polypeptide. Here's its N terminus. We have serine, alanine, alanine, glutamic acid, glutamine, serine, tyrosine, 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 and then the C terminus. Now that primary sequence is then going to determine the way in which this molecule folds back on itself and thus how it functions. So let's think about why that is. Well, secondary structure is sort of the first level of folding. Secondary structure has nothing to do with the R groups. It's only interactions along the polypeptide backbone. And in particular, it's hydrogen bonding between carbonyl oxygens and amino hydrogens. The carbonyl oxygen will have a small delta G, negative delta G, and the amino hydrogen will have a small delta I said delta G. I meant a delta negative here. And the amino hydrogen will have a small delta positive or a partial positive charge. Now these two are too close to one another. Where's my pointer? Too close to one another. But if once we spread them out about 4, 5, 6 amino acids along, they can wrap back onto themselves. When they do that, they form two common shapes. The alpha helix that you see on the left and a beta pleated sheet that you see on the right. And if you go along there and look at the alpha helix, for example, where it's twisted around, and you've got 4 or 5 amino acids in between every single one of those amino groups that are hydrogen bonded to a carbonyl oxygen. Now, which of these two is favored is still under investigation. You know, what conditions give you a beta sheet versus an alpha helix. But in general, it has more to do with the overall sequence and the way the rest of the molecule is folded, rather than the sequence of which amino acids are embedded within the alpha helix or the beta sheet itself. Alpha helices are really common in transmembrane proteins, for example. They're also common in DNA binding proteins, proteins that have to interact with DNA. The alpha helix is real good at helping proteins to do that. The beta sheets are involved in a variety of things. As one example, there are proteins involved in Alzheimer's disease that appear to be really rich in beta sheets. So there's research into trying to understand how these flat surfaces seem to play a role in the fiber formation that is characteristic of Alzheimer's disease. So when we talk about alpha helices and beta sheets, we're talking about these secondary structures that are due to hydrogen bonding along the backbone of these polypeptide chains. Alpha helices, DNA binding, membrane spanning, beta sheets can also form functional surfaces. I didn't mention that. You can have all R groups of one particular category on one side and R groups of another particular category on another. And so you can create a surface that's maybe all positively charged, or a surface that's all hydrophobic, or a surface that's highly polar or highly negatively charged. And then again, they become important in trying to understand the molecular phenomena involved in certain diseases like Alzheimer's disease. Tertiary structure, then, a little 3' that you've got down there at the bottom, is three-dimensional folding due to interactions between the functional groups on the amino acid side chains, in other words, the R groups. So when we're talking about secondary structure, it's just hydrogen bonding along the backbone. Tertiary structure, however, is all sorts of interactions between R groups that can be distant from one another on the polypeptide chain. So imagine this long purple snaky thing is a polypeptide chain, and what they're showing you are a few different types of R group interactions, and you can see how it induces folding. So here we've got a hydrogen bond between a carboxyl group and a hydroxyl group that draws this region together. Here we've got a hydrophobic interaction where hydrophobic R groups, nonpolar R groups, are going to try to hide together and away from water. Here we've got an actual covalent bond. This is a special one we call a disulfide bridge. And it turns out that when two cysteines are allowed to come together, they can lose their hydrogens and leave a covalent bond behind. We call this a disulfide bridge. Of course, this is going to be a very strong interaction. It sort of staples or rivets this long chain together at that particular point. And there are also plenty of opportunities for ionic bonds. So we see really a lot of these major types of bonds and interactions being involved in making sure that the protein folds very, very precisely so that it can carry out the function that it was designed for. Last but not least is quaternary structure, or 4'. This is where we have 2 or more folded polypeptides that need to come together as subunits to form a functional multi-subunit macromolecule. We would call that quaternary structure. We used to think that that was pretty uncommon, but it turns out that may actually be more the rule than the exception, where individual folded polypeptides don't act alone, but they are a subunit of some larger molecular machine. Now, primary structure is simply determined by the recipe. It's determined by the gene. Secondary, tertiary, and quaternary structure are determined by the primary structure, but for them to fold properly, we have another set of proteins called chaperones. Think about what a chaperone is, right? You went to your freshman winter formal, and mom and dad are sitting out in the car waiting for you. They're chaperones. There are chaperone proteins that guide other proteins to fold properly during the process of translation. And so these chaperones are essential for making sure that secondary, tertiary, and quaternary structure all happens in the appropriate manner. We've talked a lot about different types of bonds, talked a lot about folding. It's important to understand that the three-dimensional structure of a protein requires all these different kinds of bonds for all this folding to take place, and it begins with the primary structure, and it goes all the way through secondary, tertiary, and in some occasions, like this immunoglobulin G molecule you see here, there's quaternary structure where there are multiple subunits coming together to produce a very large macromolecule that can then carry out its function. That wraps up the end of our discussion of the amino acids and the polypeptides. So I hope you learned a lot, and I hope you're willing to go back and rethink through these and make sure that you understand them well. Let me know if you have any questions at all.